So welcome everyone. For those of you who have been here in past years, I try and make sure that my talk is a little bit different each year. And as in other years past, I will focus on trying to introduce some of the scientific talks that you will hear tomorrow and on Sunday to give you a preview of what they're about and what the key things you need to know ahead of time for them. I'm from the University of Toronto in Canada. I've been a Prion researcher for over 20 years, and not only am I a researcher, I'm also a family member. My father had sporadic CJD, and we lost him when I was 10 years old back in 1990, back in the early days when still not a lot was known about prion disease. And so that's one of the things that motivates me and keeps me going and makes me excited about going into the lab every day. So Dr. Appleby already talked about some of the basics of prion disease, and I'm going to restate a few of the most important points just so that hopefully it sinks in. And so prion diseases affect both humans and animals. Other neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, as far as we know, those are mostly unique to humans. But prion diseases affect animals as well. And studying animal prion diseases is in many cases much easier to do. And therefore, a lot of what we know about prion diseases has come from studying diseases like scrapie in sheep or chronic wasting disease in deer, elk, and moose. And certainly the ability of prions to cross between different species was highlighted by the transmission of mad cow disease to humans in the form of variant Creutzfeldt-Jakob disease. Dr. Appleby already mentioned the human prion diseases, and I just want to reiterate that prion disease in human is unique. That it's the only disease that I'm aware of that can occur in three completely different ways. Sporadically, meaning that there's no known infectious source, no known genetic cause. Genetically, in the case of FFI, GSS, and familial CJD, and infectious in the diseases like variant CJD and Kuru. So just a quick overview of high school level biology to get us warmed up here. So we have our genes, and our genes are made up of DNA, deoxyribonucleic acid. DNA, through a process called transcription, gets turned into another related molecule called RNA, ribonucleic acid. And then these big machinery in our cells called ribosomes take that RNA and translate it into proteins. And proteins are the major workhorses in our cells. They do lots of things. They can be enzymes that catalyze chemical reactions. They can serve a structural role to give our cells their shape. They can be involved in communication between different cells. And so proteins start out as a sequence of amino acids. You can think of them as beads on a string that then fold up into a defined 3D shape. So here on the left is the nucleic acid sequence, the DNA sequence of the prion protein. So we better memorize that quickly because there will be a test at the end. So DNA is a sequence of A's and T's and C's and G's. That gets converted into messenger RNA, which is pretty similar, except the T's become U's. And then that gets made into a protein of which there are 20 possible. The alphabet is 20 letters long. So 20 different amino acids get strung together, and that protein then folds into a specific shape. And it's the shape of a protein that defines what it does. And as Dr. Appleby mentioned, we are still not 100% sure why we have the normal prion protein, PRPC, but this is what its structure looks like. So I like to think of things simply. So this is my sort of reductionist approach to what is prion disease. Basically, it's a disease of protein structure. So you have PRPC, the normal version of the prion protein, misfolds into the disease-associated form called PRPSC or PRP scrapie, and then this causes disease in our brain. And the reason why prion diseases are especially bad is that PRPSC is self-multiplying or self-propagating, meaning that when PRPSC encounters a normal prion, PRPC, it causes that PRPC to convert into another copy of PRPSC. And this is why prion diseases are infectious and can spread both within the brain or, in the case of, say, the infectious prion diseases, from peripheral sites of the body into the brain. So conceptually, I think the best way of thinking about prion diseases is to think of PRPC as a Chevrolet Corvette. PRPC, Chevrolet Corvette, is a dynamic entity. You can do subtle changes to its structure. You can raise and lower the hood. You can raise and lower the roof. You can open the car door. But you can still tell that even with these changes made, it's still a Chevrolet Corvette. It's a car, right? When you get prion disease, you get conversion of PRPC into something that's very different in structure. You get conversion into a dangerous shape of a molecule, and this is the transformer analogy that Dr. Alpleby was referred to. I feel that there's going to come a point in time when nobody's going to understand this anymore because transformers is a product of people in the 80s and 90s, I think. But the analogy is appropriate. You have a transformation of something that's normally shaped into something that's a different shape that's bad. Here's the same thing except slightly more scientific in nature. So the structure of PRPC, you've seen it a few times now. It's on the left of the slide. Those red ribbon-like things are called alpha helices. So during prion disease, the red alpha helices get converted into structures called beta sheets. Those are the purple things in the structure in the middle. So whereas the structure of PRPC is rather globular, meaning that it's sort of spherical, PRPSC is very flat. It's planar. And so lots of different copies of PRPSC can stack on top of each other to form aggregates. And these aggregates deposit in our brain and cause havoc with our brain cells, eventually causing them to degenerate. Now, this initial misfolding of PRPC to SP or to PRPSC can occur randomly or spontaneously, which we think is the origin of sporadic CJD. It's possible that it happens in everybody's brains, but the brain recognizes this as something that's bad and gets rid of it before it becomes a problem. It can happen due to mutations in PRP because of the mutation changing not the fold of PRPC, but making it more prone to misfolding into PRPSC. Or in the case of infection, prion infections, it can be initiated by a pre-existing source of PRPSC. So just to put it a little more simply, you have PRPC as a blue circle turning into PRPSC. Then when another molecule of PRPC comes along, it binds to PRPSC, gets converted into a second copy of PRPSC, and then this can repeat in a cyclical sort of fashion. So now you have sort of two ends of the molecule. You can add two molecules of PRPC, they get converted into PRPSC, and so on, until eventually you get sort of small aggregates that are sometimes called oligomers, and current research suggests that these smaller aggregates are actually the worst type of aggregates that cause more damage to brain cells and maybe be more infectious as well. And then these can get longer and longer until eventually you get things called fibrils that are really the species that deposit in the brain. Now, how does one distinguish between PRPC and PRPSC? Because if as a researcher, we want to be able to know, is our sample, does it contain prions or does it not contain prions? Or if we've treated something with a drug, how did the levels of prions change? Well, the easiest way is to use this enzyme called proteinase K. So this enzyme's job is to chew up other proteins. So it encounters a protein, it chews it up into tiny little stretches of amino acids that get recycled in the cell. So PRPC, it's a small protein. It's a monomer, meaning it only exists as a single copy. It's soluble. And so PRPC can chew this up no problem. However, when you get PRPSC, because these fibrils are really long, it's like if you tried to put like a quadruple cheeseburger in your mouth or something like that. The protease can't quite access the parts of the molecule it needs to be able to chew it up. And so PRPSC is resistant to degradation by this molecule called proteinase K. And so that's the most commonly used biochemical method of determining if a sample has prions in it or not. You can also do these assays where you take advantage of the fact that prions are infectious proteins and they can act as seeds to initiate the disease process when you inject them into a laboratory animal. The two most commonly used animals for prion research historically are mice and hamsters. Hamsters were used a lot initially because they get disease relatively quickly after you inject them with prions, about 2 to 3 months later. Whereas with mice, it takes more like 4 to 5 months. Mice are easier to work with in a laboratory environment, and now most of the research is done in mice. So this shows you what you get when you perform that proteinase digestion test. So on the left, you have brains from a healthy mouse. So these this brain extract does not contain prions. So here, this is what's called a Western blot. It's where you use an antibody. An antibody is another type of protein, and its specific job is to go and search out a very specific protein and bind to it. So what you're doing here is looking at specifically the prion protein amongst a mixture of 1,000 or so other proteins in a complex mixture like a brain extract. So on the left, you have the healthy mice, and in the single you see where that PRPC arrow is pointing to, that's PRPC. If you treat it with proteinase K, all that signal goes away because PRPC is susceptible to digestion by proteinase K. On the right side is a brain from a prion-infected mouse. As you can see that there's signal both before and after proteinase K digestion. And those 3 bands that you see on the right side, that's PRPSC. Now, if you take a brain of a mouse that's been infected with prions and you cut slices of it and you look at it under a microscope, you can see what the hallmarks of prion disease are neuropathologically. And so if you stain with an antibody to PRP, this is the little brown dots in the image. This is PRPC that's deposited as fibrils in the brain tissue and cause it to degenerate. And the major sign of prion-induced neurodegeneration is that the brain becomes has holes in it. It becomes is filled with what are called vacuoles or spongiform change. So it gets this Swiss cheese or sponge-like appearance. And that's relatively specific for prion disease. Now, I told you that you can do these prion infection experiments, but prions are not universally infectious, which is generally a very good thing for those of us who study them. And so if you take mice and you can try to infect them with not mouse prions, but prions that exist in other species like hamsters or deer or humans, it doesn't work very well. And this is because of something called the species barrier, which means essentially that prions from species A typically do not transmit efficiently to species B. Of course, there are exceptions to this. And on Sunday, actually, you will hear about tomorrow in the keynote lecture from Dr. Glenn Telling, you will hear about all the strategies you can use to bypass the species barrier in mice. For instance, if you want to study chronic wasting disease prions in mice or human prions in mice. Now, mice and hamsters themselves are sort of somewhere in the middle on the relative susceptibility to prions. At the far left side of the spectrum, you get animals that are thought to be more or less resistant to prion disease, and those are your horses and your dogs. And this is all due to specific changes in that amino acid sequence, the beads on a string of the PRP from those different species. On the right side, you have my favorite animal. If you've been to this meeting before and seen me talk about my research, I'm the bankful guy, as the Astrocasters always introduce me as each year. And so bankfuls are an animal that's atypical because they don't impose a species barrier. They're susceptible to prions from all species. And it's all because of the unique properties of the prion protein that they produce. Now, Dr. Appleby talked about variation in prion disease. And one of the main reasons you get variation, both in animals and in humans, is because not all prions are alike. You can have what are called different strains of prions. So when you think of a strain of a different type of infectious agent, like a virus, you think of strains of the flu, which is why you have to get a new flu shot every year, because something about the DNA of the flu changes, causing it to have slightly different properties. Well, in prion disease, you don't have nucleic acids involved in the infectious particles. It's all protein-based. And so when you have strains of prions, it's caused by having different shapes of those PRPSC aggregates. So one molecule of PRPC can't just misfold into a single bad shape. It can actually misfold into several different bad shapes, each one of which causes a slightly different prion disease, typified by differences in the clinical symptoms, their molecular properties, and one of the most commonly used ways of differentiating between different prion strains is to use this protease digestion assay, proteinase K. Here are different types of human prion disease, and by the different patterns you get after proteinase K digestion, that can indicate that you have a different prion strain involved. Now, I talked a lot about or I've already spoken about how you can inject prions into the brains of a mouse, and that's going to cause that mouse to get prion disease. And this is really how 99% of the research is done in this field, because it's such a great animal paradigm. In fact, it's far, far better than the animal paradigms that exist for any of the other human neurodegenerative diseases, like Alzheimer's disease, Parkinson's disease, the tauopathies, because these mice actually get authentic prion disease that is, for all intents and purposes, identical to what occurs in larger animals or humans. So it's a great model. The one problem is that most cases of human prion disease do not originate from an infection. And therefore, some of the earliest events when that PRPC is initially misfolding into PRPSC, potentially druggable or treatable events, may be missed in this type of model. And therefore, there's been a desire to create models in which mice develop prion disease spontaneously, as would occur in sporadic CJD or the genetic CJD. And doing this is much more difficult. And one of the speakers tomorrow, Dr. Mastriani, has done a lot of beautiful work in this field. And one of the talks you'll hear about is from one of the recipients of the postdoctoral fellowship research grant, Surabhi Mehra, using the unique properties of that bankful animal I talked about to study the earliest events in prion disease in genetically engineered mice. Now, at the end of the day, a mouse is a mouse. A mouse is not a human. Sometimes when you're wanting to understand human prion disease, you need to have a human prion, or you need to have a human setting. And doing experiments with humans is obviously not so straightforward. So the technology now exists that you can take skin cells from people, you can turn them into stem cells that can then be turned into all the different cell types in the body. And if you treat these the right way, you can turn cells into brain cells that will then assemble into these little structures called organoids, or mini brains that are at least somewhat reminiscent of a human brain. And it turns out you can do prion infection studies in these cells. And you're going to hear from Dr. Ariel Hay, the other recipient of the postdoctoral fellowship, about how you can take skin cells from people who have CJD and turn them into mini brains and see what happens. Now, Dr. Appleby already mentioned RT-QUIC. I'm going to mention it briefly because one of the talks tomorrow is going to use this. So it's a great test. Unlike the organoid experiments and the mouse experiments, this can be done on the orders of hours as opposed to months or years even. And so in this test, you take PRPC, a source of PRPC that's produced in bacteria, and you apply to it a little bit of PRPSC. You use this molecule shown on the bottom left, which binds to PRPSC and becomes fluorescent. And then on the top left is what a typical RT-QUIC assay would look like if you're comparing a sample from a prion-infected mouse brain to a healthy mouse brain. So in a healthy mouse brain, you have no prions, and therefore the fluorescence signal, which is the black line, never goes up. In a prion-infected mouse brain, after about 5 or 6 hours, the signal starts to go up as more aggregates are formed, and you know that that sample is positive. So in humans, most of this test is now done using CSF samples, but lumbar punctures are not entirely non-invasive, and so we're always looking for better ways, less invasive ways to get samples from people. And so you'll hear from Dr. Matthias Schmitz talking about using tear fluid for the diagnosis of CJD using the RT-QUIC test. All right, in the last part of my talk, I want to focus on specifically therapeutics, because I think that is a topic that's very meaningful to everybody in this audience. So going back to that simple chemical equation for prion disease, PRPC goes to PRPSC, which causes disease. There are several potential ways one could potentially intervene. One way would be to reduce the levels of PRPC, because if you don't have PRPC, you can't make prions. This is a very popular approach because it would work with all prion strains. We know that it's likely to be safe because from studies in animals show that not having PRPC in your brain appears to not cause any problems. Another strategy would be to inhibit the conversion of PRPC to PRPSC, or to stimulate the clearance of PRPSC, or to block the toxicity of PRPSC. And maybe in reality, we might need to use strategies that are mixtures of these different strategies. So I'm going to talk most about the strategy of reducing PRPC because it's one of the ones that's being pursued the most. So going back to this biology lesson, you have DNA being converted into RNA, into protein. PRP messenger RNA then gets translated into PRPC protein. So two of the most the therapeutic strategies that are being pursued right now are ways of blocking this. So antisense oligonucleotides. This is the Ionis clinical trial, and there are several representatives from Ionis, I think, who are going to be at the meeting and will be speaking during the drug discovery panel on Sunday. So antisense oligonucleotides, or ASOs, bind to PRP messenger RNA and prevent it from being translated, which means that you get less PRPC being produced. Zinc finger repressors, these are being developed by Sangamo Biosciences, also a representative present in the drug discovery meeting at Sunday. These are like big fat things that sit on the DNA and prevent the machinery from getting access to the gene that is needed to make the messenger RNA that then gets translated. So this also reduces PRPC levels. And Eric Minikel, who will be at the drug discovery panel, and Sonia Valab have been exploring CRISPR-based technologies that do similar things, the end goal of reducing PRPC levels. But there are other more outside of the box approaches to doing this, and several of the grants from this past cycle focus on that. So I don't think it's been mentioned yet that PRPC is a protein that sits on the surface of cells. It's kind of like a flag sticking out from the surface of the cell. And the conventional wisdom is that PRPC needs to be at the cell surface for it to be converted into PRPSC. And so what you'll hear in the talk from Gerald Schmidt-Olms, that PRPC, because it sits on the surface of the cell, normally interacts with several other proteins that also exist on the surface of the cell. And if you take one of these such proteins and you use a chemical that inhibits, he found that that causes PRPC to be internalized into the cell and degraded. And if there's no PRPC on the cell surface, you don't get prion disease. So you'll hear about that strategy from him. Also, you could prevent PRPC from ever getting to the cell surface in the first place, which is the strategy that Gate Biosciences is pursuing, who's also part of the drug discovery panel on Sunday. Now, you'll remember that we have this wonderful enzyme called proteinase K that degrades PRPC, all of it. So why can't we just treat cells with PRPC to get rid of, or PK to get rid of all the PRPC? Well, the problem with that is that proteinase K digests all proteins, and so you would be digesting all of your proteins away, which would be game over for your cell. And so, but there are more specific proteases out there. And so one that's being pursued by Dr. Qingshang Kong is a protein called atomate. So atomate, instead of non-specifically chewing PRPC up into small pieces, cleaves it at one specific spot to produce this smaller version of PRPC shown on the bottom of the slide called PRPC1. The C1 fragment is thought to not be able to be converted into PRPSC. And this procedure also decreases the total levels of PRPC, so it has double benefit. So in his talk, you'll hear about how he's using viral vectors to increase the levels of atomate in the brain to try and prevent prion disease. Now, the best strategies for reducing PRPC levels are not going to be 100% efficient. There's always still going to be some PRPC left, and we know from studies in mice that even that will allow prions to replicate and cause disease eventually. And so we need to pursue parallel strategies. So in the strategy of inhibiting conversion, you can use something called a dominant negative prion. So if you remember this schematic shown here at the bottom right, I showed this earlier where you have PRPC adding on to PRPSC to make a fibro grow. If you use what's called a dominant negative PRPC, these bind to the ends of a growing PRPSC molecule, but then for themselves cannot be converted into PRPSC. So they act as basically stop signs that prevent prions from growing anymore. And you'll hear exciting new research from Dr. Joaquin Castilla, how he's using these dominant negatives and viral vectors to try and stop prions from progressing in the brain. Now, last but not least, preventing this toxicity of PRPC is another way you could do it. Now, studying the toxicity of PRP as opposed to the replication of prions in a simple cellular system has been challenging, but the Aguzzi lab in Switzerland has developed this really nifty test where you can take slices of a mouse brain and you can culture them like you can normal cells. You can expose them to prions, and after a little while, the cells in these brain slices will start to die because of the prion replication. It's called the organotypic slice culture assay. And you'll hear research from Elena DeCecco looking at how this molecule called glycoprotein non-metastatic melanoma protein B, GPNMB, is a potential protective factor that can block this toxicity associated with prions. Now, I just want to end with something that Dr. Appleby alluded to. I call it the propagation of the prion principle. So the prion principle is where you have PRPC changing its shape into PRPSC. PRPSC is self-propagating or self-multiplying, meaning it can catalyze its own replication, and PRPSC causes disease. Turns out that general idea is applicable to several other neurodegenerative diseases, including ones that are much, much more common than CJD. So if you forget about the concepts of infectivity for a second and think if you have a normal protein that changes its shape into a misfolded protein that polymerizes into aggregates, stimulates the misfolding of the normal protein into more misfolded protein and causes disease, this idea actually applies to Alzheimer's disease, Parkinson's disease, ALS, the tauopathies. In each disease, it's not PRP misfolding, it's a different protein that causes disease. So alpha-synuclein in Parkinson's disease, A-beta in Alzheimer's disease, TDP-43 in ALS or Lou Gehrig's disease. And because of this, a lot of the tests and ideas that were pioneered in prion disease can be adapted for use to study these other more common diseases. Some examples are RT-QUIC, first developed for amplifying prions in Byron Coey's lab. That test can now be used and is becoming the most commonly used way to confirm diagnosis of Parkinson's disease of alpha-synuclein aggregates. Similar tests are being developed for the tauopathies, such as Alzheimer's disease and A-beta. This has really revolutionized how we study these diseases. We can take small bits of protein aggregates, inject them into the brains of mouse models to study how other types of protein aggregates spread within the brain and to the brain. And the idea of strains enciphering disease variability. Turns out that occurs in other diseases as well. All of the different tauopathies, like chronic traumatic encephalopathy, which is the disease caused by getting lots and lots of concussions, or Pick's disease, or progressive supranuclear palsy. Each one of those is associated with a different shape of tau aggregates. Again, this concept was first postulated using the pioneering basic science research in the prion diseases. So one of my take-home messages I want to instill in everybody is that although prion diseases are rare, they punch far above their weight class in terms of the effect that they can have on our ability to diagnose, understand, and treat other human neurodegenerative diseases. And this is why funding prion research, especially by fundamental research, but clinical disease research as well, is absolutely essential, not only for being able to eventually cure prion disease, but for being able to cure and better diagnose these other diseases as well. So with that, I will end, and now Dr. Appleby and myself are going to stand up here and answer any questions that you may have about what we presented or anything else that's on your mind about prion diseases. Thank you very much.